The overall goal of this method is to stimulate optically controlled substances during flow cytometric measurements.
This novel device can help answer key questions in the field of cell signaling by combining the power of optically controlled substances with flow cytometry.
For the first time, optogenetic tools can be characterized and analyzed using flow cytometry. No comparable established method existed until today.
To begin, suspend transduced Ramos B lymphocytes at a concentration of 1 million cells per milliliter and transfer them to a glass fax tube. Next, exchange the black O-ring of the flow cytometer with a rubber O-ring and apply silicone grease to create a better seal. Insert the glass tube into a flow cytometer and place the LED prototype around the tube with the lights off. Measure the fluorescence intensity of DRONPA in the GFP channel.
The most critical step of the procedure is handling of the glass tubes. Glass fax tubes are very fragile, and hence it is recommended to practice the insertion of the glass tubes into the cytometer prior to doing experiments.
Then, switch on the 500 nanometer LED light on the outside of the tube and continue measuring the fluorescence intensity of DRONPA. Increase the intensity of the LED lights until DRONPA fluorescence intensity decreases in order to estimate the minimal light intensity needed in order to measure the photo-switching of the DRONPA linker DRONPA protein. Next, switch on the 400 nanometer LED light of the prototype at the outside of the tube and continue measuring the fluorescence intensity of DRONPA.